So it is my great honor to introduce Professor Randy SCHEKMAN to give an IAS Nobel lecture at this symposium. Professor Randy SCHEKMAN received his PhD in biochemistry in Stanford University. He is now the university professor in the Department of Molecular and Cell Biology at UC Berkeley and an investigator of the Howard Hughes Medical Institute. At Berkeley, he developed genetic and biochemical approaches to study eukaryotic membrane traffic. The research carried out in SCHEKMAN lab revealed genes and proteins, as well as the underlying molecular mechanisms that regulate the secretory pathway and has been widely taken reference. Particularly, Randy uncovered the COP2 machinery that regulate the export of cargo protein out of the ER, which we have heard a lot of talk in this symposium. And this is the first step in the secretory pathway. Professor SCHEKMAN is a world-renowned cell biologist and one of the founders of the modern cell biology. Among his awards are the Albert Lasker Award in Basic Medical Research and the Nobel Prize in Physiology and Medicine in 2013. Professor SCHEKMAN is a member of numerous honorary societies, including the National Academy of Science and the Royal Society of London. He has previously served as editor-in-chief in PNAS and eLife, and currently he is the chair of the Scientific Advisory Board, Align Science Across Parkinson's, ASAP. I think for most of the audience here, Randy does not need any introduction. But as a previous postdoctoral fellow in Randy's lab, I should say that it is such a great time and honor for me and for all of other SCHEKMANites to be trained in Randy's lab. Everything I learned in Randy's lab turns out to be extremely useful to establish a lab as a principal investigator here. So here I want to show a photo. Yeah, in this, this is a symposium I think Liz organized 10 years ago. Like it's a gathering of SCHEKMANites 10 years ago, around this time, I think, in Berkeley. It's such a memory experience. You'll see a lot of people in, in this photo, in this conference also, yeah. So I also want to say that, so Randy is very successful among his trainees, there is around, Randy can correct me if the number is wrong, 6 people win the National Academy of Science member, got them, yeah. And 2 of them win Nobel Prize. So including David Julius and David Baker, who win Nobel Prize just recently. So Randy, can you tell us who will win the 3rd Nobel Prize in your lab?
Someone named David.
So without further ado, let's welcome Randy to give us a lecture. Thank you.
Okay. Well, good afternoon, everyone. We've had a vigorous meeting already, and so I'll provide just a little brief interlude for the first part of my talk to give a retrospective on my career. Yusong asked me to do that, but I also wanted to talk about new results, so it'll only be partly old news and hopefully some good new news. Well, again, I want to thank Yusong and Jiawei for organizing this, for including me in this, and for doing all the heavy lifting to make this possible. I know, having organized such a meeting, it's a lot of headache raising funds and getting the room and getting help. So that all went on before the successful meeting that we've had. Well, let me start then. I was raised in Southern California, where I developed an interest in microbiology. I had a toy microscope when I was maybe 12 years old, and I remember going to a local dry river and picking up a jar of pond scum, and I brought it back to my room, and I put a drop of this on a glass slide, and I looked into it, and I just couldn't believe what I saw, this life, this proliferation of life that you all have seen, but you don't see with your naked eye. Paramecium swimming around, rotifers crawling around. This was, just an endless fascination for me. There were no academics or physicians or educators in my family. One evening at the dinner table, I was waxing poetic about all these things that I was seeing, and my father thought I was crazy. So I was kind of offended at his attitude. So I used to babysit and mow lawns and deliver newspapers, and I decided to save $100, which in 1962 was a lot of money. So I saved up all my earnings and I put it in an envelope in the closet in my bedroom. But I could never quite get to the $100 that I needed because my mother kept borrowing the money to buy groceries. So one Saturday morning, I finished mowing a neighbor's lawn, and I worked myself into such a frenzy that I rode my bicycle from the neighbor's house to the local police station, and I told the duty officer that I was running away from home because my mother was stealing my money, and I couldn't get my microscope. So they called in my father, and there were some words behind a closed door with the captain, and he came out. He was quite angry. But I learned a valuable lesson that afternoon because we drove from the police station to a pawn shop, a secondhand store nearby, and I picked up this pride and joy of my youth. This is a Bosch & Lomb student professional microscope, which I used every year in an annual science fair. And the rest was really almost charted by the path that I started in high school. I had the benefit of tremendous mentors all along, even in high school, all the way through. I had the same biology teacher who stayed with me through my entire career, and even up until the morning of the Nobel announcement, he sent me an email saying how much he was thrilled to still be alive to see this day. I then went to UCLA as an undergraduate, and I had another great mentor, from whom I learned about studies on DNA replication. I took that to graduate school to the great master Arthur Kornberg, where I really learned how to take a complicated biological process apart piece by piece. And it was because of those mentors that I then had the opportunity to build my career at the University of California, Berkeley, where I started with the intention of studying not DNA replication, but a process of membrane assembly, which at the time, 1976, was very ill-defined. Here we have a complex, in a way, macromolecule with proteins and lipids represented in a two-dimensional fluid. Little was known in any organism about the molecular mechanisms by which cells assemble their membranes. And I decided to start with a project that I will identify in a moment. But first I want to tell you about the background to this work, which is, of course, for most of you who've been at the meeting, all something that you already know all about. Cells, just for the undergraduates here, most of the cells in our body have all of the 23 chromosomes and 23,000 genes in the nucleus of a cell. And all of those genes are expressed in cells, making proteins in the cell. But about 30% of those proteins have to be exported out of the cytoplasm. And so over 2 billion years of evolution, since eukaryotes first emerged on Earth, an elaborate machinery has developed to encapsulate only those molecules, proteins like insulin, that have to be secreted from cells. And one of the most complex and fascinating cells that does this is the cell in the pancreas, the beta cell, in which insulin is made and packaged into these granules, which house a semi-crystalline form of insulin that emerges from a pathway that I'll tell you about in a moment. We understand this process, and we did even in the 1970s, because of the tools of classical cell biology, the ability to visualize cells by either light microscopy or electron microscopy, or the ability to fractionate cells into their constituent organelles. This process, the last process in the pathway of secretion, involves an event where the membrane that surrounds a granule, such as in this case insulin, approaches the inner surface of the cytoplasmic membrane, and at a critical regulated moment, the membranes merge by a process called membrane fusion, resulting in the ejection of this crystalline form of insulin into the surrounding environment, where it dissolves and spreads through the bloodstream to the tissues that use insulin as a signaling ligand. This process of membrane fusion is repeated many times within cells, such as in the pancreas, but perhaps even most dramatically in a nerve cell, particularly here as an example, a nerve terminal, where these membranes, now called synaptic vesicles, congregate at a presynaptic nerve terminal, and in a tightly regulated manner, membrane fusion results in the discharge of chemical neurotransmitters across the gap, where in this case, at the neuromuscular junction, receptors on the other side bind to and are activated by these neurotransmitters. The process of membrane fusion at this junction, likely, even when it was first discovered, is likely similar to the process that insulin granules use to affect their secretion into the surrounding environment. But when this process was first appreciated at the descriptive level by electron microscopy, nothing was known about the mechanism. In spite of this, the great cell biologist of the mid-20th century, George Palade, shown here, seated next to his favorite instrument, the transmission electron microscope, he and his colleagues then at Rockefeller University perfected a series of techniques that allowed the intense electron beam to be used to project through thin slices of tissue that had been fixed so as to resist the radiation forces of this electron beam. And this is what allowed biologists, through the techniques developed in the Palade lab, to visualize structures in fixed tissue. We now know, because of more advanced optical microscopy, that what he and others had been able to visualize at this level of resolution really does reflect the normal situation in a cell. So it was a pioneering effort that Palade and his colleagues masterminded that led to his receipt of the Nobel Prize in 1974, where he used not just that one observational tool, but others to chart the path of movement from the endoplasmic reticulum, where newly synthesized molecules like insulin originate, into vesicles that traffic through a structure called the Golgi apparatus, a structure that had been described first in the 19th century by Camillo Golgi, but whose function wasn't really appreciated until Palade, in his pioneering work in the mid-70s, demonstrated that it was a way station along a path that led to the formation of mature granules, which discharge their content by fusion at the cell surface, or where other molecules are diverted, for instance, to the lysosome. Now, in 1974, I had just finished my graduate career, and I became a postdoctoral fellow at UC San Diego with S.J. Singer, the father of the modern knowledge about the organization of membranes, the fluid mosaic model. And just the month I arrived, Palade had come back from Stockholm to attend a meeting of the American Society for Cell Biology and to deliver a version of his Nobel lecture, where he outlined the beautiful work that led to this depiction of the pathway. And though it was deeply appreciated by the audience of cell biologists, I had just come from a very reductionist biochemical approach. And I found the results fascinating, but surprisingly devoid of any mechanistic insight. In 1974, we knew nothing about any process of mechanism in this pathway. Not a single protein molecule or lipid had been ascribed a role in this pathway. And so I resolved during my postdoctoral years to try to take a more reductionist approach to the discovery of the pathway mechanistically by changing from the usual experimental organism, then using animals or animal cells grown in culture, instead to study this process in Saccharomyces cerevisiae. So at the risk of boring many of you in the audience, the reason for doing this was, first, yeast had already been used quite substantially and successfully by molecular biologists and geneticists to understand cellular processes that are shared among eukaryotic cells. Notably, Lee Hartwell and his colleagues at the University of Washington had used a classical genetic approach to identify the genes and sequence of events in the division of a cell. We now know that the process that he revealed is fundamentally conserved. It wasn't really appreciated then, but the guess was that yeast would be a good model for those processes that happen that are constrained to an individual eukaryotic cell. And yeast is perfect to do this. It's simple to grow. It's not expensive. You could produce billions of identical copies of cells. Yeast cells don't divide in half. They start the cell division cycle by forming a bud that grows in preference to the mother cell during the 90 minutes or so that it takes a cell to divide. And when that bud reaches the size of the mother cell, a cell division plane is deposited to allow the two cells to separate, and that process can go on exponentially over many generations, provided the nutrient conditions are suitable. Now, looking inside of a yeast cell, it's much less interesting and satisfying than looking at a pancreatic beta cell, but nonetheless, there are membranes, intracellular membranes, some of which look kind of like what you see in a mammalian cell. There's a structure called the vacuole, which is the yeast equivalent of a lysosome. There are tubules largely aligned across near the plasma membrane that we now know represent endoplasmic reticulum. In other sections, you can see a double-track membrane with a nuclear envelope. But our attention, or my attention initially, when I was considering this work, was drawn to the appearance of a collection of small vesicles congregating under the bud portion of a dividing cell. These images had already existed in the literature, in the yeast literature. In fact, there was another really interesting image that I found in the literature preceding my move to Berkeley that shows a yeast cell that had been frozen and then fractured along a plane within the bilayer of the plasma membrane. And here you see this bump, which is the bud, the emerging bud, and little dimples, little craters in the bud, which likely reflect nascent membrane fusion events. So the idea then was to use yeast as an experimental tool using traditional genetics to try to find the genes that are required for the production, the movement, and the fusion of vesicles, using not insulin or mammalian proteins, but using the proteins that yeast cells need when they're growing in the environment, such as on the surface of a grape. There are a number of enzymes, glycoproteins that were known that are secreted very rapidly by yeast cells. Some get trapped in a thick cell wall around the cell. Others just get secreted into the environment. And so the quest was to look for mutations that block this process. And so, armed with those ideas, but with no preliminary results, which the NIH found quite inadequate, I moved to Berkeley in 1976 to begin my career. I was selected by the biochemistry department in Berkeley and the then chair Daniel Koshland, who became my kind of father figure and mentor for many years to teach me not only how to organize my academic life, but also how to be a good citizen of science. I valued his lessons just as much as I valued the lessons of Arthur Kornberg. Well, so the idea was to look for those genes required for the production and movement and fusion of secretory vesicles, with the assumption that the vesicles, by fusing at the plasma membrane, would contribute to the growth of the plasma membrane of the cell. And so the prediction is that any time you would cripple a gene required for this pathway, that would be a lethal event, just as Hartwell's mutations in the cell division cycle were lethal events. And the way microbiologists had devised the tools to evaluate such mutants is very simple. You look for colonies that can grow, for instance, at room temperature, but which can't grow at human body temperature, a temperature at which normal yeast cells can grow perfectly well. The molecular interpretation of such mutations is they are a change in an amino acid, perhaps on the surface of the protein, that causes it to be thermally unstable, such that the protein can function at room temperature but lose its function, unfold at least partially, when the cell is warmed to 37 degrees. Very simple procedure that had been used for decades to study essential processes in bacteria and then in yeast. We applied to yeast, and by we, I mean my great good fortune was with one of my first graduate students, this then young man by the name of Peter Novick. He's shown here assaying for enzymes that are secreted by yeast cells. Note the long hair. We all had long hair back then. He has virtually no hair, like me, but on the other hand, he is now the George Pilati Chair of Cell Biology at UC San Diego, where he's had a very successful career and is one of the people that Yusong mentioned who's become a member of the National Academy of Sciences. So Peter and I embarked on a procedure, first just looking at a bunch of randomly isolated temperature-sensitive colonies, assaying individually for those cells that when warmed to 37 degrees, accumulate two different secretory proteins inside the cell. And then we looked for those cells that, when returned to the permissive temperature, would discharge the enzymes that had accumulated inside the cell. Around the end of 1978, after having been at Berkeley for a year, we had the first such mutant. It was really exciting. It had all the physiologic properties that we expected. We called it SEK1. And several months later, George Pallotti came to Berkeley for a couple of honorific lectures. And so I'd met him when I was a graduate student. He was very close to Arthur Kornberg. But I had the opportunity to tell him about our work, which was very exciting because he didn't realize that yeast cells secreted proteins. That evening, the graduate students organized a dinner on his behalf. And during the dinner, Peter told him about his work and the characteristics of the SEK1 mutant. And Pallotti said, well, now, of course, you must cut thin sections to see what's going on inside the cell. Naturally, this is something we were going to do, but we raced ahead and did it. And the great moment in my career came in just a minute with an observation that I'll tell you about. But in the course of doing this, Berkeley is the home of student protest. And when word got out that we were going to kill living organisms, a hue and cry arose. And the protest included a specific mention of our work. Yeast have feelings too. We made a successful appeal to the Animal Subjects Committee, and we've since killed trillions of yeast cells without a scream. Okay, well, armed with the approval, Novick then called me one day, I think in the summer of 1970, actually it was the summer of 1978, down to the EM lab in the basement of our building to show me an image that really had never been seen in yeast before. Recall the image I showed you a moment ago where there were only a small number of vesicles clustered in the bud portion of the cell. In this case, the field of cells was filled with images such as this, where we now know thousands of such vesicles accumulate within the cell. We know that these vesicles included at least one enzyme that we knew was to be secreted from cells but had accumulated inside the cell. We knew that these vesicles would then be discharged when the cells were returned to room temperature, so it was a thermally reversible mutation. We didn't know if these were vesicles at the end of the secretory pathway or at the beginning of the pathway. So for the next 18 months, with a lot of frenzy in the lab, we repeated the isolation. We devised procedures to enrich for mutants. We repeated the isolation and found hundreds of additional mutations that block different stages in the pathway. And then, many years later, I'll summarize with a slide from about the mid-1980s with an image such as this, indicating a pathway of secretion very much like what Pallotti had described with the pancreatic Eisner cell, but importantly, populated with genes such as SEK1 and many more that we showed to be required at the very end of the pathway. Genes required for a bidirectional flow of vesicles between the ER and Golgi, some going forward, some going back. A set of genes required to promote the polypeptide translocation event that we now know describe the channel that Tom Rappaport has described so beautifully in some detail. And subsequently, former postdoctoral fellows Tom Stevens and Scott Emmer in their own laboratories devised procedures to define the genes that are required to divert proteins from the Golgi apparatus to the vacuole. So it then became possible, since this was really still early on, it became possible to clone these genes by recombinant techniques. But for the most part, the genes that we cloned and sequenced showed no homology to any database then available. Subsequently, when the human genome was sequenced, these genes showed up as equivalents of human genes. And so we now know that this process was evolutionarily conserved over at least 2 billion years. And I use this image to show the commonality in biology. We can really, at a molecular level, compare the cells in this gentleman, Charles Darwin, to the cells of a cluster of yeast cells growing on the surface of a grape. There is a significant evolutionary conservation of the essential genes that are required to promote all aspects of cell growth and division between yeast and humans. Of course, humans are more complicated. There are many more genes. There are many more copies of these genes. But at the core level, the pathway is conserved. Well, this commonality that emerged from our work in the early 80s stimulated in part the growth of a local biotechnology company that I had the pleasure of consulting, a company called Chiron. Their idea was to use yeast as a platform for the manufacture of significant quantities of clinically relevant proteins. And so I helped them in two projects. One was to introduce the gene then available, the gene for human insulin, into yeast, harnessing signals that yeast cells would recognize. And now, many years later, 1/3 of the world's supply of human recombinant insulin is made in giant fermentation vats in which these yeast cells are grown, as an example shown here. The same company took the gene for the hepatitis B surface antigen, that's a membrane protein. They incorporated that into yeast, and they found that the antigen would accumulate in vesicles within cells, obviously without any infectious nucleic acid. Probably many of you in the audience have been vaccinated against hepatitis B. Well, guess what? You've been injected with vesicles that accumulated in yeast cells expressing the hepatitis antigen. So these experiments with a practical application were nothing that I ever had any intention of doing in my lab. But the point is that there is a fundamental connection between basic discovery and application. And this kind of joint effort between academic science and biotechnology has flourished around the world to the betterment of mankind. Now, let me turn then for the rest of my talk to tell you about work in my lab since then. But before I do that, I want to just give a general appreciation to the efforts of all those people in all those years who've been in my lab and made these discoveries. I am not a religious person, but I somehow feel I've been blessed with talent that I really don't think I would have had anywhere other than at UC Berkeley. And you heard about the success of some in their own career, but I'm proud of all of them, including the 6 current and former postdoctorals from my lab who are here with us today. So with that indication of where I really had the greatest pleasure, I want to turn to work in my lab over the last dozen years or so. Over the years, our work turned from yeast to studies on mammalian cells, largely now human cells, growing in cell culture, with a particular interest in how cells manufacture vesicles for export. So cells manufacture proteins for export, but it turns out all cells, even bacterial cells, pinch off vesicles that are shed into the environment. In a sort of a typical eukaryotic cell, this could be a mammalian cell in culture, there are 2 broad pathways for the production of such extracellular vesicles. They may be shed by budding from the plasma membrane. This is by analogy to the process of viral formation, COVID, SARS-CoV-2, HIV. These viruses bud by pinching from the plasma membrane. Uninfected cells do this too. They are called microvesicles or shedded vesicles. Another path is taken by membrane proteins that follow through endocytosis to be captured into a structure called a multivesicular body. They do so by being pinched into the interior of an endosome, and then they collect inside of this structure. This structure may deliver its content to the lysosome, where these structures are degraded and amino acids, sugars, and nucleotides are recycled for reuse. Or, discovered about 25 years ago, some subset of these vesicles fuse at the plasma membrane to deliver a subset of extracellular vesicles called exosomes. Over the years, we've studied this process of capture of molecules by exosomes, and we found that certain microRNAs are captured rather selectively into these vesicles, and in doing so, they invoke a series of RNA binding proteins, 2 of which we've described for particular microRNAs. And so the question is, what are these vesicles doing if they capture certain microRNAs? That is the major question in the field that I think has not been adequately addressed. If you follow the literature in this field, the claim in virtually all papers about exosomes or extracellular vesicles is that they are vehicles of intercellular communication. The implication being that these vesicles capture certain molecules, they are internalized by target cells, and by a process that would require membrane fusion, they discharge their luminal content into the cytoplasm or into the nucleus. That assumption has not been tested adequately. And there'll be a poster here today, I think, or tomorrow, from my postdoc, Songyan Zhang, that shows that we have failed to find really good evidence that these vesicles can deliver any content. Indeed, in the literature, there are other examples that exosomes can be taken up by endocytosis, but they either hang around in the endosome or finally delivered to the lysosome and ultimately degraded. There is very little documentation of an efficient delivery of any cargo molecules. And the reason we think for this to be true is that in order to deliver something efficiently, there has to be a membrane fusion catalyst. That's how envelope viruses work. They have evolved a fusion catalyst to specifically deliver nucleic acids into the cytoplasm by fusion of the virus either at the cell surface or after internalization into an endosome. But in fact, if you look at proteomic databases of isolated exosomes, you'll find nothing obvious about what could be promoting this membrane fusion reaction. There may be certain avenues of traffic that are efficient, but by and large, we feel they're not terribly efficient. These vesicles may be signaling ligands that activate cells at the cell surface. They may disperse small molecules as they traverse through the body, but they are floating around in our bodies by the billions for reasons that really aren't understood. So I'm going to tell you now about two studies done by a terrific team of two graduate students in my lab, Justin Williams and Jordan No, who asked a fundamental question about what are the forces, what are the conditions that promote the production and secretion of extracellular vesicles and exosomes. To do so, they've taken advantage of a tool that was developed by a lab in Japan. I'll get to that in a moment, but first I want to show you that you can actually separate the two populations of extracellular vesicles on a buoyant density gradient. This is an experiment we published several years ago from a former graduate student, Moraima Tomosh Diaz. If you harvest, in this case, it's a breast cancer cell line grown over a couple of days. If you harvest the conditioned medium and you concentrate the vesicles and then put them on a flotation gradient and separate according to buoyant density, you can resolve two populations, one of low buoyancy, marked by certain proteins that are unique or others that are shared, and a high buoyant density population, marked by the content of a force-spanning membrane protein called CD63. In studying the composition of these vesicles, Moraima and others in the lab were able to show that they contain a highly selected subset of microRNAs. And so the question is, what are those microRNAs doing? Now, in order to study the production of these vesicles, Justin and Jordan devised a very simple approach, which was to rely on a fusion protein integrated into the genome, where at the C-terminus of CD63, this Japanese group introduced a copy of N-luciferase. And the prediction is, because of the known topology of CD63, that the luciferase would be in the lumen of any extracellular vesicle. Now, we know when you grow cells in culture that some cells lyse and membranes fragment. We wanted an assay that would uniquely detect only the sealed vesicles with latent luciferase activity. And so what they did was they included in an enzyme assay a membrane impermeable inhibitor of luciferase and a membrane permeable substrate, such that one would only see the internal sequestered latent form of luciferase. In fact, you can show this in the following experiment. If you look at just the culture supernatant without including a membrane impermeable inhibitor, you get a certain level of luciferase activity. If you include the inhibitor and the membrane permeable substrate, that level is reduced about 25 to 30%, representing pieces of membrane or fragments of the protein. And yet, if you then include a detergent in the presence of an inhibitor, you see nothing. So that's a very significant baseline measurement. So in real time, over the course of hours, you can measure, using this assay, linear production of exosomes that are secreted into the growth medium. Now, for reasons that will become apparent, Justin had a quite interesting idea. He suggested that what is known about the process of plasma membrane repair, as we discussed in a presentation by Dr. Zhu the other day, I mentioned in the discussion of his presentation that it's known from work primarily conducted by the laboratory of Norma Andrews, now at the University of Maryland, that the lysosome is mobilized from the cytoplasm to patch lesions in the plasma membrane that are introduced by toxins or other noxious agents. That patches up the membrane, and a seal can be repaired. Justin imagined that the multivesicular body, which is very similar, in fact, just an immediate precursor of the lysosome, might be mobilized to the plasma membrane by similar forces. And if that were so, if the multivesicular body was mobilized to help lysosomes patch breaches in the plasma membrane, there would be a byproduct of a burst of secretion of exosomes. So that prediction, a very simple prediction in their assay, allowed them to do the following series of experiments, which I'll go through one at a time. In the upper left-hand panel, they used cells that were treated just briefly with a calcium ionophore, ionomycin, and observed a 30 or more fold increase over a very short period of time in the secretion of exosomes, dependent on the presence of extracellular calcium. More damaging, they used a protein, a bacterial toxin, Streptolysin O, and as its concentration is raised, in the presence of calcium in the medium, they were able to observe, in this case, about a 20-fold increase in the secretion of exosomes. With a more gentle procedure, by just pipetting cells through a narrow gauge needle to apply stress to the cell surface without any obvious damage, they observed a possibly twofold increase in the secretion of exosomes. Again, the secretion depended upon the presence of calcium. Vesicles that are secreted under these stimulated conditions sediment to the same high buoyant density that I showed you a few minutes ago, characterized by the position of CD63 containing exosomes. Now, there is a vast literature going back over 100 years that describes this process of wound repair in sea urchin embryos. Here's a paper from 1917, sea urchin egg protoplasm spilling out when a needle punctures the plasma membrane. Here at a lower mag, you can see a burst of cytoplasm spilling out of a lesion, just like we saw from Dr. Zhu poking a needle in C. elegans. And yet, with time and the presence of extracellular calcium, the wound is repaired so well that the egg can be fertilized and go on to produce a mature sea urchin after development. So this process of repair, as I indicated, has been studied by many laboratories, principally by Norma Andrews. And we know a great deal about the mechanism and the snare proteins that engage in the fusion of lysosomes. And we know that the damaged membranes can be internalized and degraded in the lysosome. We also know that these cells not only discharge lysosomal content, but they shed vesicles at the point of rupture. And we'll see an example of that right here. So here's an experiment that Justin did. Let's see if I can get this movie. These are cells, tissue culture cells, where all the membranes are labeled with a hydrophobic dye, and with a laser ablation at the point of rupture, you see a lot of damaged material being shed and spit out into the medium. Finally, within a couple of minutes, the gap is repaired. You'll see another example of that in a moment. We wanted to know what membranes might be, what proteins might be mobilized to either the plasma membrane or multivesicular bodies during such an insult. And so this is an experiment that was conducted by Jordan. That's a very simple one, as I'll illustrate. He used an epitope-tagged molecule on the surface of the endosome and an antibody against that epitope to pull down that organelle and lysosomes from a crude lysate of cells. With beads alone and no antibody, very little is pulled down. With beads and antibody in the presence of calcium and additional cytosol, quite a number of proteins are pulled down that are well beyond the background. If the sample is if the sample accumulates proteins that bind in a calcium-dependent manner, and if they're washed off by the addition of excess EGTA, then the signal largely disappears. Those bands that were uniquely retained in the sample that was incubated in the presence of calcium and antibody-bound beads were extracted, excised, and analyzed. And a series of calcium-binding proteins, annexins, that Andrews and her colleagues had already described to be required for lysosome mobilization were detected, as well as another calcium-binding protein called copine-3. What Jordan then did was to ask whether these proteins, one of them, annexin 6 and copine-3, are required for the stimulated secretion of exosomes dependent upon the damage by ionomycin. And so in this result, he used two different shRNA constructs to ablate the expression of annexin 6, very efficiently doing so in both cases, and likewise two different shRNA constructs to ablate the expression of copine-3. In a control experiment done without ionomycin treatment, there's a basal level of secretion independent of the presence or absence of these proteins. However, after 30 minutes of ionomycin treatment, a substantial reduction in the secretion of exosomes is seen in both cases when these two proteins independently are removed by shRNA treatment. Well, let me then summarize Justin and Jordan's results, which we published last year, with the following simple model, which is basically borrowing from what Andrews and colleagues had described for the mobilization of lysosomes. We predict that any condition that ruptures the plasma membrane or renders the cell permeable to calcium will allow the recruitment of annexins and copine to these organelles, both the lysosome and the MVB. And as a consequence, they are then, and we visualized and published it last year, these organelles are attracted to the region of damage, or the plasma membrane in general, where by fusion, exosomes are released. So from this, we draw the conclusion that the exosomes may or may not have some function, but we believe, and I'll try to amplify this in the next series of slides, we believe that the normal forces of shear that cells and tissues experience in the course of normal living, and particularly in vigorous physical exercise, may account for the abundance of exosomes seen in all of our body fluids. For instance, it's known and was published years ago that samples of blood taken from an athlete before and after vigorous exercise show a substantial increase in the appearance of CD63 in serum from a patient, from a normal person after vigorous physical exercise. There's a lot of damage to muscle cells that occurs in vigorous physical exercise, and we believe that the appearance of CD63 is a consequence of the repair of that damage. Now, we also wanted to know about what happens to the vesicles that are shed by budding from the plasma membrane. We know, for instance, and it's been published for some years, that platelets in the blood that participate in coagulation, involving quite a lot of stress in the platelet cell, secrete both exosomes and an excess of these shedded vesicles from the plasma membrane of the platelet. We wanted to know if this was going to be an explanation for tissue culture cells as well. And so what Justin did was he used the density gradient that I showed you a little while ago that was devised by Moraima Timosh-Diaz, having labeled the cells under conditions where one could quantify the abundance of proteins that appear by proteomic analysis in low density and high density fractions. The high density fractions shown on the left of this volcano plot include CD63, a protein that we know to be enriched in exosomes. And the low buoyant density vesicles have all of the full range of all the annexins, for the most part enclosed within the vesicles. the annexins have been mobilized either from the lysosome or the MVV to the plasma membrane. And at the point of rupture, at site of damage, we believe vesicles are shed by a pinching mechanism from the plasma membrane. And in doing so, they capture all of these annexin proteins that were part of the mobilization process. Here is an example of that. Now, this is a case of a cell that's been labeled with M. scarlet tagged form of annexin 2. So you see its localization largely in the cytoplasm. And when this cell is targeted with a laser ablation event, you see the same kind of massive shedding event from the plasma membrane. You can't see these particles, but they're being spit into the medium, followed by a relaxation and repair of the plasma membrane, and the cell can resume. We can show among the vesicles that are secreted, as you'll see in the next slide, that you can resolve, again on a buoyant density gradient, the high buoyant density vesicles that are characterized by CD63, and the low buoyant density vesicles separated by a few fractions here that are characterized by several different annexins. These vesicles, once they're collected, can actually be immunoprecipitated with an epitope tagged CD63 so as to capture the CD63 containing vesicles, and little, if any, annexin is co-precipitated with CD63. That is, they represent a completely distinct population of vesicles that can be resolved by this procedure of immunoprecipitation. It was also possible to use a tagged form of annexin, and the same, tagged with, in this case, with luciferase, and to use the same assay that I described at the outset of this part of the talk, where we include a membrane impermeable inhibitor of luciferase and a membrane permeable substrate to evaluate the secretion of annexin-containing vesicles. And sure enough, after addition of streptolysin O at around 200 nanograms per mil, there's a substantial increase in the secretion of annexin-containing vesicles, representing the shedded vesicles that I've shown you in that image, along with a corresponding increase in the secretion of CD63-containing vesicles. And you can see this over a range of concentrations, all of which are sublytic. That is, the cells don't die under these conditions except after prolonged treatment. So finally, let me just summarize these two studies, the second of which is now in revision for publication, and I can assure you not in Cell, Nature, or Science. We believe that the normal forces of living, not including even vigorous exercise, subject many, if not most, of the cells in our body to stress forces. For instance, a fluid shear of a level approximately the same that was experienced in the experiment where they simply pushed cells through a narrow gauge needle. That level of shear is a condition that applies to white blood cells in flowing in circulation, particularly when they're circulating through the heart and pumped vigorously to the different tissues. Cells migrating across endothelial, epithelial layers are also stretched and stressed, likely eliciting this kind of export process. I mentioned platelets, which, when they engage in blood clotting, secrete buckets of exosomes and shedded vesicles, and muscle contraction, normal muscle contraction, or that experienced during vigorous exercise. Now, having said this, though I think this could explain all the vesicles that are in our body, I can't speak to all of them, and there may be some avenues that really are involved in efficient transmission of information that would require uptake and fusion. But what remains is for someone to describe the membrane fusogen that is responsible for that event. There is only one membrane, typical viral-like membrane fusogen in the human genome. It's a membrane protein called syncytin, which Songyan Zhang has investigated in the lab. Syncytin is typically only expressed in the early embryo. When the pre-implantation embryo elaborates a layer of trophoblast cells, those cells become a syncytium, a fused mass of cells, because at that stage, the two genes for syncytin are turned on, and those proteins are at the cell surface of these trophoblasts, which allow the cells to fuse, and that syncytium gives rise to the placenta. at which point the genes syncytin 1 and 2 are not expressed normally in the body. So in a normal situation, there would be no syncytin available to promote the efficient discharge of vesicle contents by internalization from cells. Except, it turns out, and this is part of what Song-Yen has shown, tumor cells, which have latent retroviral, latent retroviral genes, tumor cells turn on the expression of syncytin. And so those cells do have the capability of fusing under certain circumstances, and you can talk to Song-Yen about that. So it may be that exosomes that are produced by tumor cells may capture enough syncytin to be functional in delivering cargo molecules. This is something that we are exploring in the lab now. Well, let me conclude, as I always want to do, by giving credit where credit is due. Not only have I been blessed by very talented and energetic and collaborative students and postdocs over many years, I still have some in the lab, and here's the crew from several years ago. I've talked about the work most recently of Justin Williams and Jordan No, many others contributing other work. Song-Yen, whose work I mentioned at the end, have made this a very happy place for me to have my career. Thank you.
Thank you, Randy, for the wonderful talk. So now we go to the question and answer section. So, Ge-Liang.
Hey, Randy, really nice to get everything together. So I'm just curious, since it's an next is sensing calcium, but why is sensing there's calcium from outside, but not from like ER, triggering like other related ER stress events? But
could be. It could be the experiments that we did all required extracellular calcium. But you're right. Maybe there's some event that's elicited by calcium stores in the ER. I don't know if anybody's looked at that.
So you mean that maybe ER stress can also trigger extracellular vesicle release?
Well, it may be that ligands that stimulate discharge of stores of calcium from the ER may do likewise. I don't know of an example of that, but it's certainly a good idea.
Can I comment on that?
Sure.
So the calcium concentration that is required for an exon to respond is much higher than you ever get in the cytosol, in the range of
the normal,
500 micromolar or something in that range at the minimum.
Millimolar, and that's because when you add a ionophore, it just comes pouring in.
Right.
So that probably requires a great deal of damage. What kind of concentration of calcium can you achieve from intracellular stores? Just micromolar?
Micromolar.
So it may not be adequate. That's a good point. Yes, Joey.
Randy, that's really inspiring talk. But I do have a question about the secretion via exosomes as compared to vesicles. So the exosomes actually are spit out, and then the membranes are not recycled. So in that sense, it's a more expensive process than the secretion vesicles because you need to constantly supply the membranes.
Yeah.
So,
but these are vesicles that would otherwise be dumped into the lysosome anyway. So they're going to be gone one way or another.
But on the flip side is that it may be a way for cells to secrete membranes. Certain lipid species. I was wondering, if.
Yeah. So Justin and Jordan and a number of people in the lab have collaborated on a review article that we're going to publish in the Annual Review of Biochemistry, that speaks to the possibility that this whole process is involved in cellular homeostasis, not merely as a byproduct, but actually, restoring some homeostasis to the cell.
Randy, that was a wonderful talk and really such a fantastic story. And I love the picture of you with the microscope. The whole thing was just totally awesome. I think there's some story for specific RNAs being in exosomes also. And can you say anything about, is there some selection there or what's happening and how are they ending up in what's the physiological?
So we published several papers on that subject. Yes, there really is a very high fidelity sorting event that we can reconstitute in vitro. Certain microRNAs, depending on the cell line, are enriched, we estimate, 1,000-fold.
Yeah.
However, their chemical concentration within the exosome is vanishingly small. They are a minor constituent of the other RNAs that are typically found in exosomes. Most of the RNAs are transfer RNAs or yRNAs or Volt RNAs. there's one paper published some years ago that showed that a particular microRNA may be present in 1 in 100 or 1 in 1,000 vesicles. So simple arithmetic would compel one to argue that it would take a mountain of exosomes to deliver enough microRNA into a target cell to have some functional control of gene expression, since they would have to work stoichiometrically. I think it's just not plausible, in spite of the claims in the literature, not plausible to suggest that this would have some role in a target cell. Now, it may be that the donor cell wants to get rid of certain microRNAs, and that may be the reason that it's happening that way.
Yeah, beautiful talk, Randy. And I don't say that very often.
Why do you whisper that, Tom? You don't
I
want to
say
get
a
a bad
lot.
reputation.
I had a question about why is it that, I mean, there are secretory lysosomes in some cells that are professional secretion fusing with the plasma membrane, but multivesicular bodies are not normally fusing with the plasma membrane. Is it because of the high calcium concentration, abnormally high concentration that they do? Or
Well, people have, I mean, it's not just what we say, but there are other papers that show that MVBs can fuse at the plasma membrane, but this is greatly stimulated by conditions that mobilize calcium. So I think it's a consequence of the fact that the MVB membrane is very similar to the lysosomal membrane, and so the two are just naturally mobilized together.
But lysosomes also don't fuse normally, right? So it's very...
They are when cells are
except for
damaged. They are when, you know, Norma Andrews and others have shown that if you use, you know, streptolysin O, suddenly you see LAMP1 appearing on the plasma membrane. It's coming from the lysosome.
Maybe it's the high calcium concentration.
Must be. It must be you know, there has to be extracellular calcium for this to work, and it's, you know, in the range of millimolar, so.
Randy, very inspiring. My question is, you're saying the membrane fusion, to be efficient, it should be protein guided. There's some protein. But I wonder if membrane components such as lipid will play a role. So for example, in polarized cells, you know, how these vesicle nodes go into one direction but not the other? Is it always protein?
Those are, okay, let's think about this. Those vesicles have the opposite topology. That's a cytoplasmic fusion event. I'm talking about a fusion event that requires the extracellular leaflet of the bilayer. And, you know, snares aren't oriented in the right way to do that. And in any case, it would be in an environment that doesn't have the ancillary factors for fusion. So it has to, it could, well, you're right, there could be unusual lipids that promote fusion, but that hasn't been, no one has, the problem is that in spite of the claims, you can now survey the literature, there is no example really where someone has measured quantitatively efficient delivery of cargo content from an exosome taken up by a target cell. There clearly is evidence that this happens, but the efficiency with which this happens is, in most cases, very low. And people have gone to the trouble of repeating these experiments in cells that are expressing VSVG protein. There you can produce exosomes that abundantly fuse and deliver their content, 100 or more fold more than without the expression of VSVG.
I'll just say that this question is very relevant in the Alzheimer's field because people are saying the tau or alpha-synuclein transmission. One potential mechanism is that this protein, aggregate protein, gets secreted in vesicle. Yeah. But how does it take it? Well, you know, imagine it can take it out by the other cell, but you're saying how does it get to the cytoplasm?
Okay, well,
to see the other protein.
Well, stay tuned tomorrow. My current postdoc, Shenji Wu, will describe our experiments on that. We find that alpha-synuclein, Yihong Ye has shown this too, alpha-synuclein is not secreted in vesicles. It's unconventionally secreted, perfectly soluble. So you're right, if it were secreted in exosomes, that would just, you know, that wouldn't answer the problem. There still has to be a membrane fusion event. You know, viruses all have some protein catalyst of fusion. There are no viruses that just have unique lipids without a membrane, without a protein fusogen.
Hi, Randy. Thank you for the brilliant talk. I'm just fascinated by how the exosomes are ejected through like through the wound. So I was wondering, what is the driving force that's moving exosomes or MVBs to the wound?
They're probably. I think we showed microtubule involvement in the delivery of lysosomes. Others have looked at this for lysosomes. I think we also found this for MVBs. Yeah, they have to be targeted on a cytoskeletal tract.
Yeah, and on top of that, is it specific motors? What is preventing the microtubules to deliver other vesicles? Why it is MVB specific?
I don't think, we have not looked at that, but I'm pretty sure that people who've looked at lysosomes over the years have not only identified the snares, but they probably identified the motor proteins, and I just don't remember which are invoked.
Thank you. So any
questions from the undergraduates? Oh, you're not an undergraduate.
Any question from the UG students?
You could, but not anymore.
Oh, okay, great.
I'm sorry, I'm a postdoc, not an undergrad.
Well, you could have fooled me.
So yeah, I enjoyed the talk very much. So I'm just curious on the last section of the shear stress. So how much the integrity? How much will it affect the integrity of this plasma membrane? Do you think like those cells have are leaky they have leaky plasma membrane?
The mildest force where we just pipette them through a narrow gauge capillary actually is sublytic. So, but maybe stretching the membrane is enough to allow leakage of calcium into the cell.
Okay, and then do you think like in future studies of like exosomes we should put cell culture on orbitals?
Well, it depends on the purpose of your. If you want to isolate more, I suggest you simply add ionomycin, and that will deliver them to the culture medium.
Okay, thank you.
There's a question in the back there.
Yes.
Maybe
there is a question in the back. Yeah.
Actual undergraduate.
Yeah, he must be another UG student.
Hi, Dr. Randy. Thank you for the talk. And I just one simple question. So, do you think this would have like a similar effect on how, for example, how nuclear envelopes would repair themselves? Because I would imagine nuclear envelope would get damaged by chemicals or toxins or whatever. So, do you think this would be a similar path of a similar way that cells could fix their nuclear envelope?
There's a whole emerging literature on repair processes of intracellular organelles. There's a fascinating process of lysosome repair that involves pinching vesicles to the outside, I think, or is it butting into the inside? To the outside. So it probably involves the kinds of proteins that you described in your talk.
Okay.
So, but nuclear envelope probably also as well. I know there is a process of a microautophagy of nuclear membranes that are degraded in the lysosome, and maybe that's part of a repair process.
Got it. Thank you.
Any more questions? Oh, here. Wow.
Okay, there's a shill as an undergraduate from my lab here.
Thank you, Randy, for a very brilliant talk. I just wonder, do you think one day we can use the exosome to deliver some drugs or some pharmaceutical agents for some cure of some treatment for the diseases?
Well, there are a lot of attempts and failures at that for reasons that I've already mentioned. However, I don't dismiss the possibility that one could engineer exosomes to be fusogenic. But I think the even more practical approach will be to simply make synthetic vesicles. And I know biotech companies are doing that. One of the problems with exosomes is it would be very hard to manufacture them in sufficient quantities to have any clinical value. So using synthetic vesicles, I mean, we already know lipid nanoparticles deliver, spike mRNA. We've all been vaccinated with that. The efficiency with which lipid nanoparticles deliver their content is not so great, though. But if you get a slug of it injected into your muscles, enough gets in and expressed to be useful.
Yeah, thank you. Because now I'm working on the phase-separated microdroplets. So I'm also curious about the exosome. Okay, thank you for it.
Okay. Okay.
So, any more questions? Here.
Oh, there's another one.
Another one
Now the
here. undergraduates
undergraduates wake up. Okay.
No, she's not.
Oh, she's not an undergraduate. You're an assistant professor. You're going to tell me now.
Right. But I'm going to ask a question. I always, I was dreaming to ask a Nobel Prize laureate when I was undergrad. I think in this classroom, so we have undergrads and graduate students and the professors at different stage, assistant professor, associate, and full professors. And I think at different stages of life, so we may have some central theme questions, like what kind of factors that drive you to be a professor, and along the tens of years of scientific pursuit, like what's the point that really make you excited and think this is the way, the right way, that I will not regret?
Well, let's see. How much time do we have left here? You know, everyone has different motivation. I can only describe what motivated me. When I started as an undergraduate at UCLA, my intention, knowing not anything better, was to go to medical school. Sorry, David. You know, I grew up in a Jewish family. What more could a son do than grow up and be a doctor? So, but when I got to UCLA, I had the amazing good luck to be able to work in a research lab in my freshman year. And the professor who took me in was himself an assistant professor, and he said, well, you don't know much, so I want you to read this book. He gave me a copy of the first edition of a book called The Molecular Biology of the Gene by Jim Watson, which was a very foundational book at the time, very different than the dusty biochemistry books that people used. And this book was, to me, a revelation. It was like reading passages from the Bible. Not that I've done that, but I had the sense of this was a new world that was waiting to be discovered. So that changed my life. You know, at that point, I just said, this is what I'm going to do. And I came home that summer. I told my parents I wasn't going to be a doctor. They were disappointed, but you know, eventually they got over it. To me, so I get lots of undergraduates who come into my lab that want to experience the lab. Mostly they want a letter of recommendation, but they come into the lab. And a subset of them can, you know, find that they have the skill set, the hands to make things work. And a further subset of them actually really enjoy that. And it's that subset that should go on in experimental science. It's not for everybody. You know, when I tell these undergraduates, look, you're going to come in, it's not like a textbook. It's not like a cookbook, you know, course, a lab course. This is real science. Most of the time, it's not going to work for lots of reasons. And you have to keep your eye on the big picture to remain excited about a goal. And if you can't do that, you will be frustrated, and this will not be for you. So the lesson that I learned as a graduate student is you have to focus. You just focus like, you know, laser vision on the big goal. You know, identify a real interesting problem that you think you can solve, and just keep your mind on that. And you know, you'll get through all the frustrations, and to enjoy the rare moments when something like that image that I showed earlier in my talk of this cells exploding with vesicles. You know, you live for moments like that. That if that gives you pleasure, if you looked at that image and got excited, science is for you.
Okay, so this exciting moments is actually the lasting driving force for the along your way till now?
It has to be. It has been for me. I still like, you know, looking at primary data and thinking of experiments and controls, and I relish those moments when some insight emerges that we hadn't anticipated. Or, for instance, when Justin and Jordan came into my office with this crazy idea about why extracellular vesicles are produced in abundance, it made so much sense, I just said, go for it.
Okay, thank you.
Thank you. So thanks, Randy. Any other question? Oh, wow, more students asking questions now.
Hi, Professor. Thanks for your inspiring talk from someone like me that not from the classical biology side. So I'm actually familiar with the computational systems biology, and I heard your idea that if we can find some virus-like proteins that catalyze the membrane fusion events, probably we can find some exosome that actually play an essential role in some biological processes. But I wonder, do you think it's a promising approach to develop some high throughput methods that sample all those exons in the fluid and check if there's any, for example, differentially secreted exosomes in certain conditions? Like, do you think it's a promising way to study exosomes?
Well, that's unfortunately, that's kind of like looking for a needle in a haystack. If you sample the extracellular vesicles that are present in blood plasma, they come from all of the tissues in your body, some more than others. It's an enormously complex set. And there might be only a tiny fraction that are actually being produced to deliver something. It would be very hard to identify that. You'd have to have a sensitive assay. So that would not be my preferred approach. Our approach is first to see if syncytin is expressed under conditions that it does get into exosomes, and if so, are those conditions that could promote the effective transfer? If there are other fusogens, whatever they are, it may be very hard to find them. We've set up an assay that is sensitive, that will detect if there are other things that promote it, but thus far we haven't had any really good results.
Okay, thank you.
So yeah, I think for the sake of time, let's thank Randy again for the wonderful lecture. Thank you.